Pleased to welcome Johannes Jaeger, or Yogi for short. Johannes, or Yogi, is very well known for his sort of deep and creative thinking about biological problems, and in particular taking seriously the complexity of biological systems and trying to find new ways of moving past sort of a reductionist or mechanistic paradigm in understanding that. So that has, of course, put him somewhat at odds with some of the mainstream, one might say. So he's also taken a very creative and unusual career decision in becoming a freelance researcher, philosopher, and educator. But recently also has gotten a big grant to pursue his work. So I guess that continues to be a sort of gravitation toward the system now that this has worked out. Anyway. Without further ado, I look forward to hearing about sort of where your scientific journey is taking you, and welcome.
Thank you very much, Jonas, for this invitation, and especially the possibility to do whatever I want in this talk as well. So I will use cells as examples in this talk, and I am a developmental biologist by training. I studied in Basel in Switzerland with Walter Gehring in a very traditional developmental genetics lab. That's one of the reasons I got into this philosophical trajectories, it's because they did these really good experiments in the lab and had the weirdest explanations for them that even as an undergraduate, I couldn't understand how they got to those interpretations. And this is something that sort of kept with me throughout my career. Jonas sort of asked me to make this a little autobiographical, but in the end, the talk didn't turn out this way. So I can just tell you that apart from this conceptual confusion, that I was always a little weirded out by explanations that my colleagues would and interpretations that my colleagues would use for their experiments. I also sort of changed fields a lot, from developmental biology to modeling, mathematical modeling, to evolutionary developmental biology, and later on now to organismic systems biology and philosophy of science, and my latest inroads are into the field of AI research. If you think after this talk biology has some conceptual problems, AI research is completely confused. I mean, heavily confused, and nothing compared to biology. It's much worse over there. So my interest is in how do we grasp the world, right? So I am a scientist by training, but I guess I've always been a bit of a philosopher, but I don't actually have any formal training in philosophy. And maybe that's an advantage. So I do give philosophy courses for scientists, but I think it's an advantage to come from the scientific angle, because a lot of the philosophy of science is not very useful for biologists. And what I want to talk about today is exactly a kind of philosophy that would be extremely useful, I think, for biologists to know about today. And the pity is that we don't learn about it during our education, and it's pretty new. So people who learned their philosophy of science earlier on, like I actually did, don't know about it. So it's basically an advertisement for this sort of philosophy of science. I also collaborate with these kind of philosophers and philosophical biologists that I introduced today. And I just would like to get you interested in maybe also chasing a few windmills, because it's still a topic that's very much on the fringes, not just of our fields, but we'll come back to that, but also of our imaginations. And so the question is, what is the use of philosophy in biology? It was Richard Feynman, I think, who said, although I don't think he actually said it, that the philosophy of science is as useful to scientists as ornithology is to birds. And I think it was Carl Bergstrom, who's an ornithologist, who said birds would actually profit a lot if they knew some ornithology. So it's true that a lot of the philosophy of science is not useful, but there is a philosophy of biology that is compatible with the latest scientific findings in our field, and is based on the best understanding of how biologists actually produce knowledge, as far as we know. That itself, if you think about it, is almost an empirical research program. To find out how we find out things is not just theoretical philosophy. It depends on the sort of conditions on the ground. But much more important than a philosophy of biology that studies how biologists work, I think what we need is a kind of philosophy that informs our own thinking as biologists to think about biological problems. And that is a philosophical kind of theoretical biology that has been around for a long time, but has been marginalized over the last, I would say, 50 years or so. And I would like to resurrect it. I still haven't got a good name for it. So if you know something better than a philosophical kind of theoretical biology, which is terrible, then please let me know. It's not very good PR. So one is sort of looking at biology and how biologists do things in an informed way that's empirically informed. And the other, that's the philosophy of biology. And the other thing I'm going to talk about today is philosophical theoretical biology. That is a practice within biology that uses philosophical tools to look at biological questions. I'll give you examples of both. So I think the reason that philosophy of, the idea that biology needs philosophy is not very popular is that over the last 60 years, we can date it pretty precisely to the discovery, the structure of DNA in 1953. Since then, and the emergence of all the technologies that came out of that, empirical progress has just been absolutely amazing in biology, and all of it was driven by theoretical and methodological advances, not by conceptual ideas. Whoops. Can you still hear me? I just froze here.
Yes, we can.
I think I'm back. Okay, so let me try and switch the slide further here, but I'm kind of stuck. Okay, we get, that's actually quite good. We get all kinds of innovations at the same time. That's how it was supposed to show. So we have technology of sequencing and various DNA manipulations ending up in CRISPR now, down to single cell sequencing and single cell metabolomics. We have microscopy, 4D microscopy techniques now that are amazing. Techniques for quantifying data, modeling data, and also we shouldn't forget absolutely fantastic new ways of culturing cells in 3D, and even moving 3D cell culture and all kinds of things that we could have only dreamed of 50 years ago. So this is extremely important because each technological or methodological advance, of course, gives you a completely new insight into nature. I am not going to deny that, okay? So the only thing I'm going to complain about is that this amazing advance and this sort of frenzy for new technologies and the frenzied development and faster development of new methods has eclipsed the sort of conceptual thinking that I'm going to advertise here, to the extent that if you ask a lot of molecular biologists today about philosophy, it's sort of a four-letter word, right? A philosophical question is just a question that you can't answer. It's not tractable. It doesn't, it's not useful to be busy with it. And so a lot of people conclude that biology has no philosophy and needs no philosophy. So this will be our starting point. Now, the problem, of course, is that this statement here on the screen, biology has no philosophy and needs no philosophy, is a philosophical statement itself. And so I'll quote philosopher Dan Dennett here, who I rarely agree with, but this quote is great. So he says, there's no such thing as philosophy theory science. There is only science whose philosophical baggage is taken on board without examination. So let's start this talk by quickly looking at the philosophy that is behind this no philosophy, only facts stance. And that philosophy, of course, is mechanicism, looking at the world as a sort of mechanism. And of course, we don't look at it as a clockwork anymore. We have moved on since Descartes first proposed this. And something I want you to know is that this whole mechanistic view of the world was invented by René Descartes to reflect the glory of God. He thought, the clocks that people were making at the time were so great that it was sort of, the universe had to work like this because God was this flawless sort of being. And we forget about this nowadays. This was the origin of our mechanistic worldview, which has evolved very quickly from this idea, very famously described in Julien Offray de la Métrise book, L'Homme Machine, got from clockwork metaphors. It moved with the technology development, to the steam engine, to chemical bodies as chemical factories, to the computer metaphors we have today. So the clockwork is still with us, but I'm going to talk about it in its contemporary sort of incarnation, which is the fact that biology is flooded with computer metaphors. And those metaphors, I will claim, are no longer useful. They're actually impeding progress in the field in a very real way. So getting a better way of thinking about biological systems is important to get past those obstacles that we've put in our own way by thinking about organisms as machines, in particular as some sort of computer-like, programs and things like that. So what I just said was most succinctly put by evolutionary biologist Richard Lewontin, who said, the metaphor of all modern science, the machine model that we owe to Descartes, has ceased to be a metaphor and has become the unquestioned reality. Organisms are no longer like machines, they are machines. And this is a very bad thing. It's okay, we need metaphors to think about, systems that we don't know how to think about. Actually, to come up with conceptual innovation, we rely always on analogy and metaphor, because there's no other way that we can expand our toolkit, our conceptual toolkit in the first place. But metaphors have sort of a lifetime where they are productive, then they become stagnant. At the end of their lifetime, they usually become actively destructive. And this is happening here. I mean, what comes with this view of the organism are the evil twin sisters of determinism and reductionism. Determinism comes in many guises. It can be genetic determinism, anything else, but we'll see when we talk about how a cell works that it's also a sort of a mechanistic determinism at the much lower level, and reductionism, of course. So here I have to be very subtle, because reductionism is only bad if it's a worldview, if you think the whole world can be reduced to some level. It is, of course, very necessary and very useful in biology if you use it in the right way at the right place. So we're going to come back to that, to distinguish those two things. Reductionism as worldview in biology, probably not a good thing. Reductionism as a method, absolutely necessary, but dangerous if it's used out of its limits. So I want to talk in particular in the first part of the talk about two theories in biology that come out of this. One is the metaphor of the genetic program on the right, and the other one is replicator theory in evolution. And since this is a cell and developmental biology department, I want to start with the genetic program. And in particular, I want to start at the very bottom. I don't even want to talk about development. This is my field where I come from, but I want to talk about how does a cell work? How can we actually think about the genome giving instructions to what's happening in the cell, right? And so we can look at the structure of a cell, for example, in this beautiful drawing by David Goodsell. These are very popular and they're beautiful. They give you an impression of how crowded the interior of a cell is. Here's a very simple bacterium, Mycoplasma mycoides, one of the most simple cellular life forms on Earth. And you see how few molecules there are in a cell, protein molecules in such a cell, and how crowded it is. And that's a good thing. But of course, this sort of drawing misses the absolute essence of what a cell is about, and that is its dynamics. A cell that is frozen like this is dead. Okay, it doesn't do anything. And so the first thing that we need to point out is that our view of the cell that is static is limited because the architecture of the cell is dynamic, transient, and fluid. So all the interactions that happen, almost all the important interactions in a cell that are above the level of metabolism are non-covalent, and they're transient and also not very specific. That's the other problem. Come back to that in a second. The very popular view of what a cell is actually made of is as a very sort of complex automaton that's built on these molecular machines that are made out of proteins. So that's Sidney Brenner's famous way of putting it, proteins as molecular machines. But as it turns out, the more and more we learn about proteins and protein complexes, they're not like that. They're more like pleomorphic assemblies. What this means is their physical state is more like a very dense liquid or a molten solid than actual rigid parts that work like machine parts against each other. Again, their interactions are very fleeting. They're very promiscuous. A lot of the non-covalent interactions in the cell are not very specific. There's a lot of sort of moonshining. There's a lot of sort of interactions that are not very specific. So cells are intrinsically extremely noisy. Most cellular processes are stochastic. Think about the way molecular motors work. So we thought for a long time that there's sort of a power stroke model of a kinesin walking along a microtubule. So it burns an ATP and then it ratchets itself forward. But what really happens is what's called a Brownian ratchet. So the molecule is driven by noise, and the noise drives it across the ratchet. So the only reason there is a biased direction of movement is because there is a constraint put on the noise that is driving the movement of the protein. So we have more and more a view of a very stochastic cellular interior. And of course, the most stochastic process, the most well studied as well, is transcriptional regulation, where we have bursts of transcription. And this idea of steady levels of transcripts that we had in the past is no longer valid. So we have stochastic bursts of transcription that determine the overall levels of transcripts. And often transcripts, especially in development of transcription factors, are very low. So, stochasticity is extremely important for the cell. And that all means, if you take that together, that there is no even if you could define instructions that are sent out by the genome to the cell, there is no predefined sequence of steps happening in a cellular system. It's, there's a lot of noise, very transient liquid, fluid interactions. And so order arises out of all of this chaos, really. I mean, not chaos in the mathematical sense of deterministic chaos, but there's a lot going on that is, sort of suddenly leading to this higher level order in the cell. And that, of course, is extremely unlike any machine humanity has ever built, right? So the metaphor of the cell as a machine, and then also accordingly of the
Looks like we've lost Yogi. Hopefully just a interruption of internet or something like that. So he will hopefully be back in a minute. Yeah, apologies, patience.
I wonder if he knows that he went offline.
I've not received any email or anything like that. Have you, Michael?
No, I don't know either. Just wait a couple of minutes.
Yeah, I guess Yogi's computer was angry about getting insulted, being called inferior to organisms. Well, we'll wait. Welcome back. We can't hear you. Your microphone, like, yeah. Nope.
No.
Now.
Yeah?
Yeah, we can hear you. We can hear you.
I'm really sorry for that. I lost my connection, and then when I tried to reestablish, my computer crashed. So it was like a worst case scenario for a talk, and I apologize for that. So I hope you didn't have to wait for too long now.
It happens. It happens. We were. I was joking that your computer got angry about being insulted, called inferior to biological systems.
Okay,
Right.
That's probably it. Can you see the slideshow again?
Yes. Okay.
So let's hope this doesn't happen again. I have no idea what happened there, and it hasn't happened before. So I did switch to a Linux machine this year, and so yeah, there are drawbacks about having left Apple for a more politically correct future. But yeah, there's always trade-offs in life. Sorry for that. So what I wanted to say is that there's no program-like there's no way that a program-like process could be running in a cell, because even if we would know what the instructions were, we don't know what they would run on. There's no distinction between hardware and software. That's one of the problems, and also there is no sequential sort of execution of steps happening in a cell. So the other really mysterious thing is that, of course, computers don't build themselves, but cells self-manufacture. This is from a paper from my friend Janny Hofmeier. So they not only self-fabricate all the components they need for their future survival, but they also assemble them in the right way, so that they continue to have the right organization that ensures their survival. So this self-manufacturing property of cells is something that is extremely different from any machine we know how to build today, although we've tried, but it's still mysterious how you could even build such a machine. And so this is where I want to quickly mention a very, very highly abstract and formalized theory by Robert Rosen about the self-manufacture of cells. And Rosen in the 1950s already came up with a really highly abstract thinking about living systems that he called relational biology. And he thought, what makes all living systems different from non-living systems? And he came up with this really, really simple drawing here to illustrate the concept that he called closure to efficient causation. His terms are very idiosyncratic, so I'm just going to call it organizational closure, which basically means, so let's look at this little diagram for a second. Its basic elements are two kinds of arrows. So you have material causation, a sort of a process. You can go from A to B. I don't know if you can see my mouse pointer if I go to it. And that sort of process here reflects metabolism, he said. So you have all the metabolites A that go into the chain of the network of reactions that is metabolism. They come up out as products B. And those are controlled by F, which you can think of as the enzymes. So they control which reactions are happening and which ones are not. But at the same time, of course, enzymes are produced by the metabolism itself. So you also have a material arrow up to the enzymes. Now, the influence of the enzyme on the metabolism is not one of actually participating in the process, but a constraint on the process. So Rosen called this efficient causation. He uses Aristotelian terms in slightly unusual ways. But you can think about the white arrows as some sort of processes and the black arrows as some sort of constraint. And he called that repair. So what the enzymes do, sort of being produced by metabolism itself, is the replenishment of the enzymes. He called it somewhat confusingly repair. So he called this a metabolism repair system. And the trick that living systems do is, he said, all the efficient causes in a living systems are accounted for from within the system. So you produce them all inside the system. So if you would go through all the arrows in this diagram, which I'm not going to do, I just want to give you a sort of an idea of the idea, is that all of those things that produce black arrows, like F, the enzymes, are themselves materially produced from within the system. So it's all a game of placing arrows. And then he used a mathematical tool called category theory to show that not only is this sort of setup of interactions different, fundamentally different from mechanisms, machines that he called them, but that it cannot actually be captured by a algorithm in the sense of something that can run on a Turing machine. So that doesn't mean that you cannot compute part of the organism, but that the organism can always do things that are not in your model. So Rosen called this, the organism has no biggest model. So you can model the behavior of an organism, but at some point it will be able to do something that you haven't included in the model because it has this weird circular causal structure that you see on this diagram. And for years, this lingered in the back of the minds of many theoretical people thinking about biology, but never connected to empirical biology because it's completely abstract and it's not clear how it really, the F and the N and the B, that's still okay, but the phi there, what that maps to in the cell, people didn't know. And so my friend Yanni Hofmeier has done some really amazing work that I cannot do justice here because I'm just giving you the high-flying sort of overview of the whole thing. Is he mapped all these arrows in Rosen's diagram to the cell, and he actually realized it doesn't work. But he came up with a better scheme, which is shown here on top, and it's a bit more complicated. So don't worry about the arrows in particular. All I want you to know from this slide is that you can map those arrows onto specific processes in the cell. And Yanni called that fabrication assembly systems, FA systems, instead of metabolism repair systems. But you can show mathematically that they're equivalent to Rosen's systems, and they map to cellular processes one-to-one. In particular, they map, they tell you that you need enzymes and ribosomes, of course, to produce anabolic metabolism for a cell to survive. You need the cellular milieu and chaperones helping it to drive non-covalent folding and assembly of protein and protein complexes. And you need transmembrane transporters to maintain the electrolyte composition of the cellular milieu. So you get a minimal model of the cell that has this weird non-computable structure of Rosen's model. And it maps to specific components of the cell, although you should notice that the cellular milieu is a systems level variable. So it also means that the organization of the cell is not reducible just to its parts. Okay, so you need at least one system level process to control the milieu to make it work. And so what Rosen is saying here, and I'm glancing over a lot of the technical details, I just hope you get these two main points. One is you cannot actually simulate the entire potential of behavior of an organism with an algorithm, and the other is that you have a hierarchical, causally circular structure, but it's not just regulatory feedback. That would just be circles in material cause in Rosen's diagrams. It's a kind of a hierarchical cycle of regulation. You can think of a cell as a holarchy, to use Arthur Koestler's word, and that is a hierarchy which has no global top or bottom. Every part in that hierarchy is both whole and a part. It makes your head hurt a little bit if you think about it too long. I think of it as sort of a series of Russian dolls, and the biggest one is contained in the smallest one again. So it's like something MC Escher would draw that goes on forever, but it's really hard to imagine this sort of stuff. So all I want you to take away from this is that if you look at the organization of a whole cell, it has a really, really weird organization that we have a really hard time thinking about, but that we can mathematically prove cannot be captured by the sort of mathematical tools we use. And this view of the cell and the organism actually goes back to Immanuel Kant, who already said each part of an organisms are special because each part of an organism owes its presence to the agency of all remaining parts, and in turn exists for the sake of the whole. Each part is reciprocally both end and means, and this is not how we build machines, not at all. There's always a hierarchical fixed organization. And if you then take this a little bit further, and again, I'm flying over a lot of details here, is organisms manufacture themselves. This is from Stuart Kaufman's book, Investigations. This gives them a degree of autonomy from the environment. So some of the causes of what the organisms does, they come from within this organization of the organism, this weird self-referential circular structure. And that means organisms are agents. They act on their own behalf. That is a scientific explanation of why an organism can have goal-oriented behavior, for example. while a machine cannot. So this has deep implications for research in AI, of course, and all that. Okay, but for us here, it means that this machine metaphor of the organism, and especially the genetic program, it just doesn't work. It cannot account for what the cell is at all, because cells and hence organisms are not machines, they're agents, and the behavior of living systems cannot be completely captured by algorithmic simulations. We have that from Rosen's proof, which is still, or conjecture, it's not a proof, that is still standing today despite some criticism that he got. And that means, and that's quite astonishing, is that living systems are processes in the real world that are not computable in a Turing machine. So the idea that everything is computation in the world is just plainly false. And this, of course, also means that brains are not computers because they're a part of the organism. But in fact, it's even worse. The behavior of living systems cannot even be fully formalized by any mathematics based on well-defined finite sets. And that's pretty much anything we would use to describe it, like dynamical systems theory. It fortunately excludes the kind of mathematics that Rosen used. So it's not a sort of a disproof of Rosen's own work. Okay, so I'm probably telling you something you already knew, that the genetic program is not a very good metaphor, but I'm telling you in a way that is mathematically rigorous. And so here we have a principled argument why it is not possible to empirically study and understand whole organisms just by looking at their parts. Okay, and that should worry us, right? But sort of we have fixed ourselves, I think it was Johannes Jaeger who said we don't even study life in the lab anymore. And that's a weird state to be in for biology, I think. But what we can definitely say is in the overall picture, the genetic program is really a bad metaphor. And if we want to understand better how cells are organized, how they behave, then we need to move past that metaphor and come up with better ones. Okay. So this was sort of the big example in my talk. The smaller one is replicator theory, which comes up in my work in evolution. And of course, that's most extremely exemplified by Richard Dawkins' idea of the selfish gene, and which was formulated in a bit more sophisticated manner by David Hull, philosopher of science, by the way. And they said the units of selection of evolution, which play the functional roles, functional units of selection play the roles of replicators and interactors in evolution. So a replicator is simply an entity of which copies are made and which transmit their structure directly and relatively intact in a replication. So that's the genes in your body. That's why genes have been picked out as special for evolution, while the interactors, that's you and me, that's our bodies that interact directly with the external environment. In such a way that replication is differential, that selection can take place. So the interactors are basically bodies, organisms, phenotypes. And so here's a diagram by John Maynard Smith from 1972 that summarizes evolution, and it's quite complicated, but the main thing is basically just that there are replicators and genes in the germline, and they are the only thing that actually gets propagated, apart from, of course, the environment on top. So the interactors, they stop, they're produced by the genome through a program, of course, and there's an environment that's also continuous but not inherited, it's external. And then selection works by filtering out certain replicators. So these are the unlucky replicators. And it's a very clean sort of sketch. It's a form of extreme Weismannism where you clearly separate causal processes of inheritance from development and make evolution tractable and simple. And it's suited to the idea that replicators are the units of evolution, right? So this perspective suggests a single coherent process of evolution through time. It simplifies the problem of evolution greatly, but it ignores the interactions, the complex interactions between inheritance, selection, and development. And then it also takes the existence of replicators and interactors for granted. I don't know about interactors, but we can look at replicators very quickly, and there are huge problems here. The first is that, of course, we've already said, or we've already covered in the first part of the talk, that there's no straightforward genotype-phenotype map, there's no genetic program. But even worse, replicators don't replicate, okay? So there's no thermodynamic way to get a naked DNA molecule to make more of itself. It's just, it's not gonna work. Where does the energy come from and all that? You can't look at that in isolation. And if they would not produce selectable variation. They would just degenerate in what Manfred Eigen has called an error catastrophe. And there would be no species or evolution at all, because no matter how low the error rate would be, it would just accumulate over time. So replicators can only evolve if they are embedded in the more complex dynamics of a reproducer process. And that is the real minimal unit of selection. There is nothing that evolves below what is basically a cell cycle that includes both reproduction and what I call ontogenesis, which is development, but also all the processes that lead to the completion of a unicellular cell cycle. So it's a very broad idea of development. So to conclude this, the completion of the life cycle requires organizational continuity. So the cell needs to be organized like Rosen said it is, all through the cell cycle. And what is propagated through the generations is not just genes, but actually, if you think about it, the metabolism in your cells has been running constantly and uninterrupted since the first living cell on Earth, the common unit, the common ancestors of all life. There is a continuity of metabolism as well, and the structure of those reactions and its dynamics have been given on from generation to generation too, and we can't just ignore that. So the replicator theory isn't doing so well either. The basic unit of evolution is not a replicator, but a reproducer, a complete life cycle, and the replicators are just embedded in that much more complex process. Ontogenesis and reproduction are both needed to produce selectable variation. You don't get it otherwise. And reproducers are not machines. They are agents. They have this organization that we were talking about before with Kaufman. They're able to act on their own behalf. This kind of agency is what produces the adjacent possible. That's Kaufman's term, the sort of emergent possibilities that come out of evolution. So if you want to deal with open-ended evolution and understand radical innovation in evolution, where novelties come from, you need to deal with this sort of much more complex theory of evolution. Okay, so this is a very concrete way. We cannot even understand how evolution generates novelty without having this switch in frame. And this is important because somebody will ask me, so what's the empirical implication? What kind of prediction does this make? The first thing it does is it doesn't make new predictions, but it gives you a completely different frame, a different perspective to think about biological problems, which allows you to ask new questions and also to get new answers, therefore. So, but immediately what it means is that replicator theory, this second sort of consequence of the machine metaphor of the organism in biology today, just goes out the window. There are no such things as mechanically reproducing replicators. You need to have this whole complex life cycle to actually reproduce them. So biology is really different from just engineering. And so the question, of course, that comes up is, how could we just go so wrong with our metaphors and not even realize it? Because we're all happily continuing with these sort of wrong conceptual frameworks, and we're having empirical success, right? Biology is producing more results and insights than ever. And that's exactly the problem, that just generating insights, empirical success, solving problems, doesn't mean you're actually understanding the context of the problem that you're looking at. So this sort of focus on technological progress is great. Don't get me wrong. Again, it has created a hugely different view of life and a lot of insight. But you know, we have a lot of data that we didn't have a few decades ago. But it has created two really serious problems that are now not just hindering our progress in biology, but also create a dangerous gap between our ability to manipulate living systems and our ability to understand them. And also those systems like ecosystems and social systems that contain them. We have a view of them as machines too, and we think we can manipulate and control them like machines. So one is the biggest problem, I would think, is that biological knowledge has been decontextualized. We think that something we cannot track empirically right now is not worth asking. And so a lot of the questions that we have in biology have been lost. And it's okay to think that, because we think that, you know, next year we'll have new technologies that will answer more and more questions, and eventually we'll understand everything about life. But I don't think that's true. We have truly decontextualized our knowledge. We don't understand anymore where these big mounds of data that we've produced are situated in, you know, the bigger picture. And that has some really not just detrimental, but outright dangerous consequences. And the other thing that happens more socially on the social level is there is a real disdain for theoretical work in biology. I mean, it's idle speculation, it's sort of wasting your time, and you could be doing experiments instead, right? So these problems are not just peripheral to modern biology, but they are existential. And that not just for our discipline, but for humanity itself, I think. You know, if you're not scared about CRISPR yet, you should be. And the sort of manipulations we can now do, gene drives out in the wild, are horrific. We have no idea what such gene drives will do, because we think about nature as a machine, right? We can manipulate and control the consequences of that. There will be unintended consequences, and I don't think they will be good. So in a way, and I'm a bit polemic here, I'm aware of that, we could say that we have never been more wrong about life than we are today. We have all these heaps of data, but our fundamental view of life as machines is wronger than ever, basically, before, because in the past, before the scientific revolution, nobody had these kind of ideas about life. The universe was a living universe. It was very different, and it was scientifically wrong. But I mean, we can think about whether our machine view is actually any better, and what kind of practical consequences that has. And also within biology, I think we urgently need to relearn to ask the big questions. So I would argue we're at a special time in history where we really need to reconsider and think again. So this is sort of the concluding part of the talk. Jonas, do I still have about 5 to 10 minutes, you think? I'm sorry for the interruption before.
Sure.
That didn't help with the time schedule. So the first thing that we need, of course, is new metaphors, right? So think about the absurdity of the program metaphor. If organisms were really like computer programs, they'd have to be able to not only write and rewrite themselves, but also to manufacture the hardware they're running on. And in fact, there is no hardware-software distinction, so it really doesn't work. If you think about it in dynamical systems terms, like I have for years, then you can treat the organism as a dynamical system, but then it have to write its own equations and generate its own boundary conditions and solve itself in the process. So that is also, you know, you always need some external methodology hardware to apply to it, but organisms don't do that. They really self-manufacture. So these metaphors don't work. Philosopher Dan Nicholson, whose work I've used before but not mentioned explicitly, only on the slide, has come up with a sort of stream of life conception of the organism as an alternative metaphor. He says that living forms are not in being, they are happening. They are the expression of a perpetual stream of matter and energy, which passes the organism and at the same time constitutes it. So unlike your car where you put gas in, it burns it and it comes out again on the other end, what you eat is your food is becoming you, right? You self-built. And so Dan thinks about life as a stream, or maybe better as a sort of a self-organizing system like a candle flame or a hurricane. But this is still limited because the hurricane will dissipate its entropy gradient as quickly as possible. And what living systems do is they do a bit more than that. They maintain the conditions for their own life much longer. And so I would suggest a conception of organisms as agents. And again, the agent is a metaphor. Agency comes from our human interactions with other human beings. But at the same time, since we humans are organisms as well, it's of course a much more accurate metaphor. But we have to be careful with not putting intentionality and anything like that in it. Agency here only means that living systems are composed of acts, in the words of process philosopher Suzanne Langer, who wrote a book in the 60s called An Essay on Human Feeling. I wonder how many of her male colleagues read that at the time, but it's actually really good. It's their loss. An organism, she says, is built up by its own acts. And that sounds a little absurd at the time, but if you think about it, there are modern forms of that, like Terry Deacon's idea that living systems are teleodynamic. They are able to maintain, not just to use entropy gradients and self-organize, but to maintain the conditions for their own existence. So they can delay the time of their demise, basically, by keeping the organization that keeps them alive going in a very organized way. So this is what Rosen's organization is all about at this very high level of understanding. I'm sorry if I go very fast across a lot of sort of very counterintuitive notions here. But I mean, the basic idea is really that living systems are agents that act on their own behalf. And that's a better view of, a better framing of biology than the machine metaphor. So why does it matter? And are just a bunch of new metaphors? They're not going to do it, right? So I think metaphors do enable an urgently needed reframing, a recontextualization of biological knowledge. So to connect all the heaps of data that we have to the bigger picture. But this kind of recontextualization can only happen if in the real world, if we actually create the right institutional incentives and structures for the right kind of theoreticians to survive and thrive. We used to have this kind of philosophical biology, and I'll come back to that in a minute, but it's really hard to survive with this kind of work in academia. That's one of the reasons that I'm not on a traditional academic career, because I really want to do this kind of science. And it also requires, on the intellectual level, a better understanding of the knowledge production process itself among biologists. We need to teach students philosophy of biology again as part of a well-rounded scientific education, because the machine view goes together with a machine view of science itself, a sort of a mechanicist's view, which I call naive realism, which is just you have a method, you apply it, you get better knowledge. And that really doesn't work. So finally, reality is much more complicated than that. Finally, we need to show that philosophy is useful for tackling specific biological problems. And we need to resurrect this kind of philosophical, kind of theoretical biology that needs a new name for our age. Okay, to wrap up, what do I mean by a naturalistic philosophy of biology? So first of all, older theories of knowledge didn't take the limited nature of human beings into account. So a modern naturalistic philosophy needs to do that. It needs to be grounded in empirical evidence gained through our interactions with the world. That's the only source of knowledge that we have, and not by some abstract analysis of scientific language or anything like that. It needs to acknowledge the constructed and fallible nature of our scientific knowledge. That's a no-brainer nowadays, I think, without, though, lapsing into some postmodern relativism or radical constructivism and say, okay, science is just another way of knowing, just like poetry or religion. That's not true. It needs to be adapted to the specific context of contemporary biological research, never claiming to provide ultimate answers or the general account of knowledge production. And it should strive to always be compatible with our most up-to-date scientific knowledge and our best understanding of the knowledge production process itself. And what I think is really important, and what a lot of people don't understand, not just in biology, but also in philosophy of biology, it shouldn't just describe biology, it should actively contribute to biological research by providing new concepts, perspectives, or by resolving conceptual confusions and misunderstandings. So in my view, this should consist of three pillars, this philosophy. One is perspectival realism, which means we can interact with reality and experience it. There is a reality out there that's independent of our minds, but we always experience it from our own perspective as individuals and societies. So this is against the naive realism that thinks that you can just apply the scientific method and then you get objective knowledge. The second is process philosophy. You need to pay attention to the processes. And in biology, this is the biggest thing, right? Everything in biology is a process. Nothing makes sense in biology except in the light of evolution. That's what Dobzhansky said, but I would say in the light of the organism and its evolution, and both are processes. And the last thing is deliberative practice, which is instead of debating and finding out who has the better theory, we have to acknowledge that many theories fit reality because theories are always underdetermined by evidence. And we need to allow for more diversity in our fields and more discussion. There's very little conceptual discussion in biology, and we need more of that. I have a whole YouTube lecture series called Science as Process and Perspective that you can find on my YouTube channel about that. And that's part of the philosophy course that I provide. Last thing, and then I'll stop, is this philosophical kind of theoretical biology. And of course, the organicists of the 20th century, and the last one of them, C.H. Waddington, were great examples of that sort of biology. Waddington's landscape gave us a whole new set of concepts to think about embryogenesis, about the stability of trajectories, creodes, of homeoresis, the stability of those creodes, and so on and so forth. He invented too many words probably, but he gave us an entirely new set of visual metaphors and concepts to think about embryology. And he was the last of the great. But in the meantime, a lot of people have survived. My own master supervisor, Brian Goodwin, or Stuart Kaufman, who I mentioned before, Terence Deacon, or Yanni Hofmeier. These are all people who do philosophical work. Robert Rosen, of course, in biology. And so they've kept carrying the torch, but it's been largely overlooked. So in my own career, and this is the last slide, just to give you a few examples, I've done, especially lately, a few things. I've worked with philosopher James DeFrisco, and we came up with an account of how people explain things in Evo Devo through mechanism, and that these mechanisms are actually processes, not machine-like assemblies of parts. Also with James, we talked about how genes cause things in complex regulatory systems, and that's not very, very simple. Of course, it's some people say it's completely impossible. So we came up with a framework, a conceptual framework to think about causality in such systems. And we have a conceptual paper on the homology of processes. How can you, for example, homologize developmental processes? For example, somatogenesis in different lineages, and so on. So this clarifies. So people say homology is confusing. Yes, it's only confusing if you allow yourself to be confused about it. And this paper is about that. With my long-term collaborator, Nick Monk, we've come up with a new concept of modularity in development. And with Stuart Kaufman, I've worked on how organisms come to know the world and how that is fundamentally different from AI. And I have 2 papers in print about agency and evolution. And that's the stuff I've actually talked about today. So I hope this is sort of useful, shows you that there's useful work to be done for philosophy in biology. So I look at biology a little bit like this sunflower field in the morning. We have to lift our heads towards the sun again and start thinking, because I think in history, after 60 years of uninterrupted technological advances, I think we're at a point where thinking is becoming increasingly important. And I hope you will be inspired to pick up on a few of these things and sort of yourself reflect on the methods you use, the concepts that you take for granted. Biological concepts, no matter what concept you think about, they're always confusing and have multiple meanings. It's amazing. So you can start pretty much anywhere. And I'm sorry for the interruption during the talk. You can find a lot more information about my publications and the courses I give and all that on my website, which is indicated below. And I'm on Twitter, which is probably one of the reasons that I'm no longer in academia. So if you want some abrasive, but hopefully entertaining commentary on a lot of issues, then you can follow me there. Thank you very much for listening. Sorry to go over time.
Thank you very much, Yogi. Yeah, I'm not on Twitter myself, but I have seen some of your tweets, and yeah, I think abrasive and entertaining are probably the right words to put there. Anyway, I'm very I'm glad you've mentioned the YouTube and your papers and stuff, because my first question was going to be, where can people go to learn more? Because I think all the things you've talked about are quite far out, obviously, from the way that we usually think about things. And I think for many of us, they are very interesting, but that there needs to be sort of a digestion and sources to start reading. So you've already answered that question. Mm-hmm. So since we're at 1 p.m., I'm going to say we're going to conclude the official part of the seminar straight away. But as I mentioned at the beginning, we have another 15-20 minutes, something like that. So for everybody interested, please do stick around, ask questions, discuss, etc. And I see we have a first maybe question coming up from Namid. Did you accidentally put your hand up?
Yeah, I did. Sorry, I was going to do the clapping hands, but I don't actually know where that's going. I need to shoot off, unfortunately, but I'll catch up with Yogi later on.
I can't see those clapping hands anyway. That's a bit.
Great talk.
Thank you.
Really enjoyed it.
Thanks.
Cheers. Bye.
Cheers. All right. Well, yeah, put your hand up if you have a question and or a point you would like to make. I guess while people still sort of deliberate, I will sort of start with something. And I mean, you've pointed out the like empirical success of the sort of mechanistic and technology driven paradigm. And that it sort of self-justifies itself, right? And I find it very, like, while I very much agree with you that it is high time now to think a bit more. Like I find it hard to imagine that actually will happen unless these kind of new ideas actually do lead into sort of empirically self-justified productivity as well. So do you, I know it's still early days for the kind of perspectives you've shown us, but do you foresee directions in which this will happen eventually?
Yes, so for the, I mean, I presented the agency stuff, which was a bit of a risk because that's still the furthest away from yielding results. But we've done this work on process homology has, for example, been used in a very recent eLife paper about the evolution of RNA interference. So this is a sort of almost like a field guide for people who work on homology and try to homologize processes. So they can use it in their work, and it's been used already. So I think these kind of things, and also I hope the dynamical modularity stuff, which we've used in our own empirical work to good use, are going to be picked up fairly quickly. Some of the bigger picture things are going to take time. So one of the messages I really want to drive home is that we can't just stick to stuff that in biology that is tractable right now. I mean, biology is so incredibly complex if we stay at the organismic level, but then if you go to ecosystems, it's getting worse. And then of course, all of social sciences. So if we stick to, we have a huge bias of just doing what is empirically tractable right now. And that is, of course, like the metaphor of the guy who loses his key, but searches for them under the street lamp. And that's sort of, you know, it's an old cliche, but it's really true for biology. We don't see what's going on around us. And we do know that there are issues in biology that are not tractable, but are very important. And I think, so what I hope to see immediately from that is a frame of mind that we need that stops treating biological systems and social systems as manipulable and controllable. They're not, and we know that from experience. There's lots of empirical evidence that they're not, and we keep on making these interventions that are quite drastic without accounting for the unexpected side effects that we get. And we have more and more power to do this, and it gets worse and worse. And you know, the whole history of human disruption of climate and the ecology and the sixth or the ninth extinction that we're in right now that you see is actually just a consequence of this type of thinking, really. So I think there are two ways in which you can become a better biologist. One is by contextualizing your knowledge better, and then the other one by producing more results. So to that, I think it will, on the long term, increase productivity if you take on higher risk, if you think in big terms. But in this system that we have, where short-term productivity or efficiency is favored over long-term productivity and better outcomes, it's problematic, and we really need to work towards changing that system, which is really thoroughly broken and is a very hostile place for a theoretician who wants to be ahead of time. Right? Good theory is ahead of its time, so it'll pick up maybe impact, but only after a while. If you're behind your time, then you're not picking up anything at all. But if you're right in the present and you're suddenly popular, then it's also not what you want, right? And so our system at the moment, our academic system, is very sort of hostile to that sort of stuff. So I am hopeful, but not optimistic, let's say.
Right. Yeah. I mean, I guess. By the way, people do raise your hands. Do jump in.
It was sort of a really high altitude flight over a lot of really difficult conceptual territory. So
we do have a question, Bella.
Yeah, hi. Can people hear me fine? Okay.
Yes.
Yeah, thank you for this really exciting talk. I enjoyed it a lot, and I agree with a lot of the comments you said about, you know, nowadays we need more philosophy in biology to kind of understand things more conceptually. I'm just out of curiosity, like you mentioned in your talk that we should probably start asking big questions again about life and about life in general. And have you given any thought into like questions like consciousness? And because you said life intrinsically is not it's not a machine and it's not computational. And a lot of people now in consciousness are studying consciousness using a computational approach. And just wanted to hear about some of your opinions on that, and if there is any like new perspectives or angles that you think would be good to understand this problem.
So the agential perspective that I presented today is going to be really useful there, because we're just writing a paper right now that the basis of cognition and consciousness are not computational. They come out of this weird organization of the living cell that goes, down to bacteria. But there's some people who claim that bacteria can do something like cognition, and I don't think that's useful. For me, cognition is sort of agency with a model of the world that can be wrong. It's complex enough to be wrong, so you can let your ideas die in your stead, sort of thing. And so on this view, consciousness evolves out of agency and cognition. So you have agency, and then you have cognition, and then at the end you get consciousness. And what it's for, I don't know what consciousness is, to be honest, and I don't think anyone in consciousness research does, but I do have a suspicion that it evolved to make sense of very complicated situations that we find ourselves in. And so that's the problem of relevance, which is something that AI famously can't do. So basically, the first step in cognition is to define what the problem is, what is relevant to your problem. And computers can't do that because they have their worlds are well defined. Our worlds are never well defined. We have a lot of sort of ill-defined problems. This is a lot of my focus right now goes into this kind of research. How do you get cognition and consciousness from agency? But then the project that I just got stays at the bottom of that before we can fly away and ask these questions. I think we really need to have a closer look at how organisms how agency plays into the behavior of organisms and then their evolution before we can understand those more complex problems. But these are really interesting, exciting questions. And I think if you want to go into that, there's a huge opportunity, because basically all of AI research and a lot of cognitive neuroscience is on a really wrong track there right now, and is having a lot of problems with that.
Yeah, I saw some theories from David Chalmers saying the problem of consciousness. I don't know if you know these things that he said. Yeah. There's the hard problem and the easy problem, and the easy problem is with computational, and hard problem really is about generation of feelings and those subjective things that's kind of inaccessible by the scientific methods. Because
so Chalmers is a dualist. Nothing that Chalmers has ever said in his life has made any sense. Few humans have been wronger than Chalmers. He's someone who sits in his armchair, doesn't know anything about the biology of agency or consciousness. He's a dualist. It's a fundamentally mistaken position, and he thinks about zombies. What if you had all the molecules in our body but you had no consciousness? It's absurd. Please stay away from him. He's a philosophical charlatan, and he's really.
This is not Twitter, eh?
No. This is dangerous stuff. He's famous. He's written a book called Reality Plus now, where he claims that virtual reality is just as real reality. This is self-defeating, existential crap. It's gonna destroy us all. Okay, it's delusional. It's really, really bad. And it's a sign of the pathologies of modern academia that someone like that is as famous as that. That's all I have to say about this person. And I know, Jonas, it's not Twitter, but this is someone who is really doing a lot of, having a lot of bad influence at the moment with ideas that are completely absurd. I'm sorry, that's all I can say about this.
It's something of a matter of perspective, but yeah, you were asked the question, so you gave your answer, right? So I think that's totally fair. Matthias.
Yeah, it's great. I look forward to the cognitive paper that you are writing. When is it?
Yep.
When are we going to be able to see it?
Hopefully, it was planned to come out yesterday. No, it should be. Hopefully, I'll finish it and put it out as a preprint before Christmas. That's the plan. And I'm sorry to have gone on a rant, but yeah, this is bad stuff. It's like political populism. You just want to stay away from it.
Yeah, I understand. It's fun to watch as well. Yeah, thank you.
Hi, really great talk. Thank you. I was just thinking that like a bit of a devil's advocate, argument is that like most scientists nowadays are sort of not doing what maybe they want. Maybe they agree with you, but they're constrained by the funding bodies who want them to produce research that's in some way useful. and applicable to medicine. And there you have the very mechanistic sort of point of view. And so I was thinking, like, if you want to sell this to a broader community of scientists, it needs to be sort of useful in that sense, that it produces new ways of sort of helping humanity, without, like, you know, something that a biologist would agree with you, but with something where the funding bodies would agree with you.
Right.
And I wonder, like, you know, what kind of sort of achievements from this philosophical perspective can you get, that, like, you know, how can I approach sort of new projects with this philosophy in mind, to really sort of advance you know, the state of the field in a way that sort of you know, that more people will agree or come with, like, support, other than, like, sort of philosophical biologists themselves.
Yes. So, I mean, first of all, I think there is an awareness among funders. So I'm busy in the open science movement as well, and they have. So out of that come a few efforts, for example, to change the way we evaluate scientific projects, right? And so that's very interesting. So they use process approaches, for example, that, you know, often good scientific projects that give insights, they fail to achieve their aims, and our current funding system is much too aim oriented. And often the kind of thing you need here is an open exploration. So they switch to actually assessing the process of a project. Was it well done, instead of did it produce the papers it was supposed to produce, which is a huge step in the right direction. So there's some awareness and some discussion at the level of the funding bodies. I mean, I don't want to draw completely black pictures. I think a lot of people are actually aware of the problems, but we're in this game theoretical trap, right? That if you stop producing, then someone else will produce. And so you have to keep on producing. It's like an arms race. And so we have to somehow, you know, get out of that. So when I give retreats for people to survive and thrive in academia, I use evolutionary metaphors, like instead of competing to get adapted in the current system, try to do niche construction and find your own niche. And I guess that's what most of my career has been about trying to carve out a niche that allowed me to do the stuff I really wanted to do. Often that included working. We had a big EU grant when I was in Spain, when we had the big financial crisis there. And it was all about developing algorithms for better model optimization. And we used our fly work as the evolution of flies as a test case for that. So you don't see that as cheating the system. I think they invested their money very well. They just weren't aware that we did something completely different than we actually applied for. The actual research was a test case in the project. And so this is part of the niche building. Always ask yourself, how far am I willing to sell my soul? And then what will I do to actually get money? And the problem is, of course, most acute if you're passionate about lab research. And I don't know the answer to that. We do need a different funding structure. Again, there are initiatives to have longer term funding for young researchers. This is crucial, right? So these sort of rolling contracts that maybe give you 10 years when you're starting out to develop something pretty early on when you're a postdoc. That would be great, right? Then people can take risks again and build something up. But at the moment, it's hard to do that. But still, the strategy I would recommend is to somehow look for that niche that you can build yourself and survive there. It's tough, but it's not impossible. I mean, the other personal advice that I would give you is that if it's really not making any sense, I mean, there are ways to do research outside a traditional academic career. It's still very hard, but it's going to get easier to pursue an alternative trajectory. But still, if you do empirical work, it's very hard right now. So you just have to sort of find a way to survive and shield yourself. And on a personal note, just make sure that you're not going crazy. And the increasing pressure is pretty terrible for young researchers right now. And you got to stay sane. It's not worth it otherwise.
Rodrigo.
Can you hear me? I'm sorry, I got.
Yep.
Well, I don't know. I guess I had a lot of questions before, but like, I just want to say that I really admire what you do, man. Like, huge respect for how you have done it. As an evolutionary biologist, I really agree with you a lot as well. I think we need to change that perspective of genes and mechanisms guiding whatever is happening in living systems, yeah, in life as a whole. Personally, I think we need to see life as you said, kind of a more fluid system, yeah, that is adapting precisely to the environment. I guess that is the key part that we are always dismissing a little bit, you know, kind of like, yeah, the organism is doing its thing. And you said at some point, you know, like we program computers, but then, you know, computers do, but like there is a context in which life does what it does always, yeah. So I think, yeah, really, really good talk, man, really good chat. And then lastly, I just wanted to ask, because you say that you are a lot in the open science and stuff, have you checked these things related to like a research hub, you know, which is this place in which from your scientific publications or opinions or reviews, then you get paid like in a crypto, this special crypto that, you know, crypto is booming right now, is one of the 1,000 projects. And then related to this same environment of like crypto and open science, there is this big movement of DeSci, decentralized science, which is also, you know, aiming to these types of processes in which the scientists get actually personally rewarded by their job, yeah, not in this mechanism of the big journals in which, you know, they take the credit and they take the money and that's it. In this DeSci and Research Hub and GitHub, the researchers get the money, yeah, straight by their opinions, their reviews, their papers. I don't know if you are already a little bit on that.
I'm not aware very much of that. I know about efforts like in, you know, if you're in bioinformatics and computer science, it's easier to get paid straight away. I don't know these efforts, so if you want to send me some information my way, that would be fantastic.
Yeah, definitely.
Yeah.
It's really cool. That's something I've been doing. Well, not actively doing, but like, you know, getting into that. Yeah, and it's very interesting. I think precisely for people that is trying to do it on the side, yeah.
So getting paid for your work is a really big issue that you bring up, and it would be amazing if we had. I mean, so you always have to be a bit careful with these kind of systems because so there was something like the what was it called, the mechanical Turk for programmers, and it often leads to self-exploitation because you're then getting paid for what you produce, and again it incentivizes producing a lot. And of course, for code where you have specific problems to solve, it's fine. But we already have way too many people publishing way too much, right? So we have to try and publish less. So I think the way to go is really to through the hiring committees, through things like that, and say we need to look at the quality, not the quantity of what people publish, and also not just superficially, you know, not just through which journals do they publish in and so on and so forth, but what you know, I asked the mathematician, how do you hire people? And they say the field of mathematics is still such that most people read your stuff before they hire you. I don't know how you do that in biology because it's so huge at the moment, but we have to have a way. Maybe Jonas, GPT, you know, AI is the way to go. I doubt it. It's just going to perpetuate our biases. But yeah, so these are interesting. So business models, Rodrigo, I'm always interested in because I haven't found the business model that really works. And I think we need alternative business models really urgently. So please send me information if you have.
Just in the interest of time, maybe, did you want to ask a final question, Francois? Or because I saw you had your hand up.
It's not Francois, but close.
Okay.
No,
well,
thanks. It was a very interesting talk. I have a question. It was very entertaining what you mentioned about Chalmers. So I'm going to mention another name, Popper, and he talked about the paradigm shift and normal science versus normal science. And he said that like after a paradigm shift, there's a lot of this mopping up that kind of like keeps the normal science alive. And that is very important because you train a generation of new scientists, and maybe like forcing a paradigm shift is not going to work. Like obviously it's very important, like we all agree. So that's kind of like this devil's advocate question. Like, is it productive to say, have a better idea now?
No, that was not my intention. So that's one of the things I teach in my retreats as well, is so creativity, and this is something that's really dangerous in our current system, requires leisure. You have the best ideas under the shower for a reason. It's when you're not stressed or when you walk the dogs, And so physiologically, we have we need this sort of defocusing as part of the creative process. There's even scientific work on that shows that, and so that's really important. So what I'm talking about is when I talk about, we haven't talked about the institutional aspect of this all, but it's really important to create structures for people to have leisure again. And I'm not talking about being lazy, just about having the freedom. If you're in a 2-year postdoc contract, you just don't have the freedom to sit around and think, and you're stressed the whole time. I know that I've been there, and so it's just a generally very bad system. So I think you can create circumstances, you can create the social and also the intellectual environments where good ideas are more likely to happen. I don't believe so. I just talked about the whole hour about how social systems are not controllable. This is not a controllable system. You can't. So this is all this talk about, we foster excellence and blah blah blah. It's all bullshit, because nobody can do that. You can just actually give people the freedom to do that, and then it happens if you're lucky. But often it doesn't, and you have no control over that. And that is something that's really hard to sell. Of course, that's a truth that even the funders and the politicians know. But I mean, in the current environment that we're in politically and societally, it's really hard to tell them about that. But we really need to sort of collectively relax and reflect more. And then, so as an evolutionary biologist, I always think if you have too little variation and too much pressure, too much selective pressure, you automatically land on a local peak of fitness, and you're stuck. And I use this metaphor a lot, and maybe we can use it with politicians and funders too, because you have these much higher peaks around you, but you need to relax the pressure on the system to get to those peaks and let people play. That's the other thing. Science is play, Science is infinite play. And if you can't play, then we're not going to innovate. And I think the system is at a breaking point right now where we will, innovation, true innovation will, PR will go up, but real innovation will go down, and people will notice. And people are already, among funders and politicians, worried about that at the moment. So I don't think it's impossible. It's just we have to give ourselves credit that it's a very difficult societal moment, historical moment that we're in, where we have a very weird system that is very machine-like in its entirety. We have a machine view of the entire society that we live in. And even we optimize ourselves all the time. And we have to get away from this. I think it's soul-crushing, and it's eventually self-terminating, because if our wisdom doesn't catch up with our power to manipulate, we're doomed.
Thank you very much. I agree that we're not being awarded for thinking, and that's wrong. So that's a very good answer.
All right. Thank you again, Yogi, and thanks everybody for your interest and sticking around, asking questions, discussing, etc.